Hello everyone. In this video we are going to continue our discussion about the animal diversity, and we are going to dig into the different groups of animals. So let's start. So we will start with invertebrates, and these are animals living without the backbone, and they represent nearly about 95% of all animal species. So although we will spend a good time with vertebrates covering them, but keep in mind they represent only like 5%. But since we are vertebrates and we are vertebrate centered, that's why we will dedicate a good amount of time for covering the vertebrate. But first we will go through the invertebrates, and as you can see, the different phylogenetic groups are shown in this phylogeny. They all have evolved from ancestral protist. So a common ancestor of all the invertebrate is a protist, and as the name indicate, protist means or pro means the very beginning. So the protist is believed to be the common ancestor of not only the invertebrate but the different groups of animals, so the fungi, the plants, and the animals as well. So what we will do, we will start with the Porifera, which includes the sponges. Then we will move to the Eumetazoa, so this will take us to the Cnidaria, and then we will move on to the Bilateral. So they have lateral symmetry, and we will start with the Lophotrochozoa and then the Ecdysozoa, and we will end this by covering the Deuterostomia. So starting with the Porifera, so this group includes the sponges. So as you can see, the Porifera is located in a separate branch in the phylogenetic tree of invertebrates because they are very simple and they don't resemble the rest. So that's why they are kept in a separate branch. And the reason is that when we go through this table, so this table is there to connect the characteristics that we have shown in the previous videos. So the four characteristics of symmetry, the presence or absence of tissues and cavities, and the type of development. So we'll see that sponges, they are asymmetrical, so there is no plane that divides them into two equal halves. They have no true tissues, they have no cavities, and they are neither proto nor deuterostome. So nearly they are none in all the characteristics. So you may be wondering why they are still classified as a group of animals, because they fulfill the definition. If you remember, we said animals are multicellular heterotrophic eukaryotes that obtain their food by ingestion. So all these criteria in the definition, they are applicable for the sponges. So how these basal animals obtain their food and digest it, although they lack true tissues. So we will not mention any digestive tissue, but how they obtain the food and deal with it. So what we will see that these are suspension feeders, so they feed on food particles suspended in water. They have what we call choanocytes. So choanocyte is this flagellated cell here, and they generate a water current through the sponge. One thing to mention about the choanocyte is that the ancestral protist that is believed to be the common ancestor for the invertebrate was very similar to the shape of the choanocyte. So you can think that the ancestor Proteus is more or less similar to the choanocyte in structure, and that is because of simplicity. So what is happening is that water is drawn through the pores, and we have many pores here in the sponge. That's why the name Porifera. So water is drawn through the pores and it goes from there to the sponge cell. So as you can see here, so water carrying the food particles suspending in it, and it carries oxygen as well, so it will go in this central cavity, and because of the movement of the flagella, this will create a water current through the sponge. So what will happen there is phagocytosis of this suspended food particles inside the choanocyte, and the choanocyte can then digest these food particles by phagocytosis. So here the food particles and oxygen are circulating in this area. The waste products are then ejected through the osculum. So this osculum, this central opening, is the exit of the waste products like the carbon dioxide and the waste food products. So the next group is the cnidarians, and cnidarians include the hydras, the corals, and the jellies. So as you can see, sometimes we refer to the jellies as jellyfish, but it is not a type of fish. So Cnidaria is the first group of the Eumetazoa. What are the characteristics of the Cnidaria? They are characterized by having radial symmetry, so you can have indefinite number of planes that divide them into identical halves. They have diploplastic tissue, meaning that they have two tissues, the ectoderm and the endoderm. Because they have two tissues, or they are diploplastic, because of that they have no cavity. So if we mention that they have coelom, this is not applicable. So the word coelom is not applicable here because they are not triploplastic. And in terms of development, they are also neither proto nor deuterostome, so they resemble the sponges in this characteristic. Moving on, the cnidarians, they are characterized by a body plan with having what we call a gastrovascular cavity. So as the name implies, gastro means digestive, vascular means circulatory. So this cavity will do both functions: the movement of food particles and their digestion and uptake, and then the circulatory function as well. They also have a single opening that function as both a mouth and anus. So here we are showing an example of the cnidarians. They are characterized by these tentacles. So here is the gastrovascular cavity, and the equivalent of it be shown here. So what is happening? They uptake their food, and we will show how they uptake the food in the next slide. So the food particles are circulating in this gastrovascular cavity. There are there are cells that secrete enzymes. It will digest these food particles, and there are other cells that will absorb the digested food molecules, and then the waste products are going to be ejected from this exit. So it acts as an entry and exit as well, so mouth and anus. We have different versions of the cnidarians. So we have a polyp, something like that. So this is a sessile organism that is going to be anchored to rocks at the bottom of the oceans. Or we have the medusa. So this is a planktonic moving or swimming version of the cnidarians. so for example the jellyfish, and how they uptake their prey, how they capture their prey. So they are carnivores, and they use the tentacle for that reason. So they use the tentacles to capture their prey, and the tentacles are equipped or armed with what we call a cnidocyte. So if we zoom in, this structure is the cnidocyte, and this cnidocyte contains certain cells or specialized cells. We call them nematocysts. So these nematocysts actually they eject a stinging thread. So as you can see here, this is a nematocyst, and as you can see, it contains a thread that is coiled. So what happens? So once the cnidarian are close enough to the prey, they release the stinging thread, and by doing that, they grab on the prey and they uptake their prey.